Hello and welcome. My name is Melita Irving. I am a trustee of the Royal Society of Medicine and past president of the Medical Genetics section. And with both those hats on today, I'm delighted to bring you the 29th episode in the RSM's COVID series of webinars for healthcare professionals by healthcare professionals. And this time we will be focusing on the role of the human genome in the coronavirus associated disease. And with me today, I'm delighted to welcome Professor Sir Mark Caulfield and Dr. Kenneth Baillie. Mark is Professor of Clinical Pharmacology and co-director of the William Harvey Research Institute at Queen Mary University of London. He's also Chief Scientist for Genomics England, set up in 2013 to deliver the 100,000 Genomes project on whole genome sequencing in rare diseases, certain types of cancer and infection, and successfully delivered, I have to say. His research interests are in molecular genetics of hypertension, so very well suited to today's subject. Kenny is a consultant anaesthetist at the Royal Infirmary in Edinburgh, and he also has an appointment at the Roslin Institute, University of Edinburgh, where he has a research program in translational application of genomics in critical care medicine. His PhD studies centered around statistical genetics in infectious diseases. So again, you can see why our two guests make a perfect combination for today's webinar. We have had lots of questions already submitted to us by those at the point of registration, so thanks very much. But as we go through, please feel free to throw your questions into the Q&A and upvote the ones that you like, because then that will prioritize them and we will get to those sooner. Without further ado, the starter for 10 goes to Kenny. So Kenny, why do we even think that the genome could play a role in COVID-19?
That's a good question to start with. So the background evidence from other infectious diseases is that the chance of a patient dying from any infection is very strongly encoded in their genes. So we've known for decades that the heritability of susceptibility to infection is stronger than for other common diseases like cancer or heart disease. And we have plenty of examples from other infectious diseases of very strong host genetic effects. In COVID, there's less direct evidence because we've only known about the disease for a few months, but from the Twins UK study, there's evidence that the disease is heritable. And from the COVID-19 Human Genetics Consortium, a global consortium of genetic studies, there are already what look like quite robust, reproducible genetic associations. So we have a good reason to think that it's likely to be important, and if it is, then it could potentially lead us to new treatments for this disease.
So, I know that you're working quite closely with Mark and in a collaborative way with others in the UK and also internationally. So what are we doing in the UK to try and get to the bottom of this genetic susceptibility?
Well, the study we're running is called Genomic, and we're working in partnership with Mark and the team at Genomics England to do whole genome sequencing on critically ill patients with COVID-19 and controls who have mild or asymptomatic illness. And this is really a UK-wide collaborative project. There was an existing network of intensive care units at the start of the outbreak recruiting to the study. We were studying the genetics of susceptibility to infection in critical care. We had focused on influenza and sepsis, but in our original inclusion criteria, we described SARS and MERS as key conditions that we would aim to study. And then, of course, when COVID started, without us really doing anything, the cases started pouring in. We've now expanded the network to a total of 205 intensive care units across the country, which is almost all of them. And through a tremendous community effort, research nurses and clinicians in all of those units have been acquiring DNA and consent from patients with critical illness caused by COVID-19.
That's great. So it sounds as though we were ready for action when this hit us. Do you think that's the case, Mark? Were we in a position to try rapidly to get to the bottom of this?
I think in terms of the genetic basis, Melita, then I think we are quite well advanced in accruing patients and enrolling them and getting an extremely good response rate. I think in terms of the overall pandemic, were we prepared? I think we could probably all say probably not. In terms of are there learnings that we could make that we would, with planning perhaps, have not had to make? I think we could all agree that there are potential things that we would do differently and better, but that's easy with the retrospectoscope. With regard to the program that we're embarked upon, which is the largest such program in the world, aiming to enroll up to 20,000 severely ill people, comparing them with 15,000, as Kenny said, who are mildly affected or asymptomatic, and using whole genome sequencing, I think we hit the ground running, and we're already sequencing, and we'll have sequenced 1,000 in the next week or two, 5,000 by the end of August. So I hope that with Kenny's help and leadership, we'll be able to make important observations that will support us in a secondary wave.
And if I may ask, what kind of things are we looking for?
So the opportunity, Kenny emphasized that actually we're looking at whole genomes. So this is probably one of the early times in which whole genomes has been applied in this way to this type of trait, and at scale. So the UK has got the largest such program at the moment. As Kenny said, there are international endeavors which we must, for the good of humanity, all join. But the virtue of a whole genome, Melita, as you know, is you can read as much of your and my genetic code as is possible to read. And that means we can find the things that make us susceptible to disease. And in the case, to the point that Kenny made, you may not have exhibited any of the features in your genome if you had not come across the challenge of SARS-CoV-2. And so we're in a unique scenario where it's very clear that people have very differential outcomes, some of which might relate to socio-demographic factors or indeed other comorbidities, but equally so, the patterns would fit with a genomic architecture for the disease. Kenny has rightly pointed out about heritability already. And I think that the final point I would make is that we already have good evidence from your and my world that there are rare causes, rare variants, that can cause severe immunodeficiencies. We've got very many good examples, particularly in children and younger adults, where we have found rare variants with major effects, but they would have probably had negligible effect if you hadn't come across SARS-CoV-2.
And I guess that kind of takes us on to the concept of the genetic background, genetic variability and susceptibility to COVID-19, and genetic variability and the severity of the disease, which are two very distinct situations. So Kenny, what can you bring from your past about the susceptibility, what genetics tells us about the susceptibility to COVID-19?
So that's a fascinating question. Mark and I and the team at GEL have had lots of discussions of that. So susceptibility, by definition, would be after exposure to the virus, you develop a disease. So any symptoms, and susceptibility to severe disease is, I think, a subset of that. And I think there is evidence, particularly for COVID, that there are different mechanisms underlying susceptibility to developing symptoms and susceptibility to life-threatening critical illness. Probably the best evidence of that comes from the astonishing effect that we saw for dexamethasone in the recovery trial, and the apparent differential effect in predefined subgroups according to severity. So I'm sure everyone's aware of the result, but there was a very strong effect in critically ill patients that was not evident in hospitalized patients who didn't have a requirement for oxygen. So the mechanisms might be different, and we'll need to consider that when analysing the results. The third component, of course, is outcome. So once you're sick, once you're critically ill with COVID-19, what are the genetic factors that determine your outcome? And that's possibly therapeutically the most interesting part of the study, but the hardest to tackle with genetics, because when we're looking at outcome, we're comparing two very similar groups of patients. That is, patients who are critically ill with COVID already, and we're comparing those who survive and those who do not.
Okay, thank you. And yes, Mark, if I could just pick up on something you mentioned about, so you and I are very well versed at understanding rare diseases and taking from what we learned in the context of rare diseases to apply that knowledge to more common complex disorders. And I think this is a perfect setting for that. So what kind of things might we learn from the rare disease effect of this condition? so when it affects people that are, we would consider atypical to develop severe COVID, that we can then apply to understanding that susceptibility in subgroups, in the more typical subgroups.
So I can give you an example, Melita, from the program so far. So the NHS is running a rapid exome service. What I mean by that is that we're sequencing the genome, but only the 1% of you and I, your and my genome, that codes for the proteins. So it's a very restricted picture, but nonetheless, it may be where major findings in rare disease could be detected. And of the first 4 patients sequenced through the National Health Service rapid exome service for intensive care, one of the 4 has a combined immunodeficiency, which has been amenable to a treatment, and that person, that child, has recovered. So there will be, as you and I know, there will be other examples, particularly in the younger people. From a recent piece of work by Professor Ken Smith from Cambridge, published in Nature about 3 weeks ago, we see individuals who possess rare variants, and sometimes rare and common variant combinations, to your earlier point, Melita, that actually, when they come across the wrong challenge, suddenly tips them over the edge and they possess an immune disorder, either autoimmune or some form of immunodeficiency. And as you and I both know, that there have been multiple children for whom we've detected immunodeficiency, and in some cases this has paved the way for a therapeutic option, and that's the ideal outcome. But I'm not going to try and persuade the audience that's going to happen for the majority. I think that would be misleading. But there are potential things, and Kenny and I have discussed this, where we may make a genomic finding that would allow us to either repurpose a medicine that exists, a bit like the Dexamethasone story, or bring something off the shelf that we've not been in use, and that could be something that the biological insights from this would unlock.
Yeah. So I guess we can look at this in 2 ways in respect of genetics and susceptibility. Is this unmasking some previously unknown immune disorder or some susceptibility through the immune system? But of course, there's also the possibility that because the virus uses the ACE2 protein as its receptor, maybe there's something about the genetics of the ACE2 that might confer susceptibility. Do we know anything about that?
Well, the thing that we do know in relation to that is that the one major study published in the New England Journal a few weeks ago highlighted a region on chromosome 3 where there's a transporter that actually modulates to some extent the ACE2 receptor. So we don't know the precise biology, and we absolutely don't know whether this is really the gene that is of interest because it's within a region containing other genes, but it's the most biologically plausible in that region. And so it's well worth looking at that first. And this is the sort of thing we can get from this. Segments of the genome highlighted where we can do additional work to refine our understanding and get closer to the biology of the disease, and then perhaps use it to inform therapeutic strategies downstream.
And one of the hot topics that's coming through on the chat actually is around the ACE2 receptor, and if studying that could give us any idea about the astonishing observation that people from Black and ethnic minority backgrounds are more likely to get more severe form of the disease. Can you speak to that? Is there anything that we know about that yet?
There's been a lot of speculation. I mean, I might let Kenny come in on this in a moment because he'd probably have more to add. But if I just say that in relation to the renin-angiotensin system, there are polymorphic variants within the standard angiotensin converting enzyme that many of the people on this seminar will have used to manipulate through ACE inhibition. And there are polymorphisms, and there are quite marked differences in their representation and the concomitant angiotensin converting enzyme activity. Now that might feed downstream to ACE2 through the rest of the renin-angiotensin system. And there was speculation at the outset of this that ACE inhibitors and angiotensin receptor blockers appeared to be preferentially consumed by people who had adverse outcomes. we now know from Lombardy that that's not due to ACE inhibition or ARBs. But Kenny, you might have something to add on that.
Well, thank you. I agree completely, Mark. The evidence currently to support a specific genetic effect there isn't coming through from the small studies that we've done so far. I think in general terms, that's exactly what we could potentially do with genetic studies, particularly identifying a subset of genetic variants called EQTLs, expression quantitative trait loci, which are genetic variants that alter the amount of a RNA or protein that is expressed in a patient. And that's a lever that's a bit analogous to the effect of a drug. So a drug alters the complex molecular cellular systems that lead to disease, and the gene alters those same systems. And in some cases, those effects are parallel. So we can use that to draw an inference about what we would predict the effect of a drug acting at a particular target would be. And as Mark said, that will help us to prioritize treatments for use in clinical trials.
Yeah, so this is moving us in a certain way towards precision medicine, and I think this has really been highlighted as we mentioned earlier with dexamethasone, and perhaps part of the differential response for people with severe COVID has been around a genetic variation in their response to dexamethasone. So this is quite an interesting field to again drive us into the next iteration of genomic medicine. So it's very exciting. We're getting a lot of, Kenny, do you want to say something on that?
Well, there's a fascinating opportunity there because we it's a before and after study, but there was a Tuesday at about midday where Peter Harvey and Martin Landry revealed the dexamethasone observation from recovery to the world. And at that point in the UK, certainly, the majority of COVID patients will have been treated with dexamethasone. So if there's a very strong effect, we'll potentially be able to detect that in the genomic study and in a parallel study called ISARIC-4C that many people on the line will be aware of and will have been recruiting to.
Yeah, fascinating times. We're also getting a few questions on blood groups and the role that some association with blood groups plays in the severity of COVID-19. Mark, do you have anything to share with us on that?
Well, we've been actually discussing this very thing. The first genome-wide study of about 1,880 or so cases from Spain and Italy, compared with about 2,200 controls, showed a striking association to the region I just mentioned, where a membrane transporter may influence ACE2. But then the second locus that was genome-wide significant was the ABO blood group. Now, we just have to be a little bit cautious about that because it turns out that the control group included blood donors. And as people on this seminar will know, blood donors sometimes are preferentially selected for their blood group because they have a rare blood group and other people need that blood. So there could be a potential confounder there. We're very keen to continue to look at that. But there was a suggestion, as you rightly highlight, and the audience do too, that there might be an association in that one study, and it was quite striking, but it does need corroboration, I feel. I don't know what you think on that, Kenny.
Yes, I agree. I mean, I think that's something we've learned over the years of genetic studies is that often the first findings can be misleading and replication is essential. I think in the case of an outbreak where the implications of any discovery could be useful immediately, and of course we'll have a time limit because the outbreak will, I hope, pass. I think there's a justification for sharing those results immediately, but we have to interpret them cautiously.
And so if we just turn our minds now to the virus and the host interaction and the genetics of the virus and the genetics of the host and how those play a role together. Of course, the virus is constantly changing, constantly mutating, and new strains are occurring all the time. What does that mean when we think about if we identify, if you like, high risk groups in the public health context? Will they remain high risk, or will that change as the virus itself changes?
Well, that's entirely possible, I guess. Kenny, what would you say?
Yeah, my guess is that that the effects that we're seeing will be fairly consistent for this virus. I mean, as Mark says, it's possible that the tropism of the virus could change, and as Mark and I have discussed in separate calls, that there are variants already in the virus that appear to alter the transmissibility, but as far as we know, not the severity of disease. I think in order to change who's getting susceptible to it in sort of broad demographic terms, I think the mechanisms of disease would have to change, and my guess is that that's not likely.
I would agree with that, and I would also suggest that we, as a country, have sequenced the largest number of SARS-CoV-2 viruses in the world, over 30,000. There were over 1,500 different introductions of SARS-CoV-2 into Britain, and there is a recent form of the virus where, as Kenny says, it's showing increased transmissibility, but that may not necessarily be associated with worse outcomes. Sometimes when the virus becomes more transmissible, it actually becomes milder. And so we don't yet know what the answer to that is. There aren't enough outcomes. But the thing, Kenny and I have been very keen on this bit. We have linked to the so-called COVID Genomics Consortium, which is COG UK, and we are going to combine the viruses and the patient's genome, and we'll be able to ask, are there within virus factors, within host factors, and then is there a combination of the two together that contribute to the outcomes? And nobody's done that before, to our knowledge, at scale. So we are linking that and doing through special ethical approval using the NHS number.
That's great news. We've got quite a few questions coming in about age as well. So we've got quite a few paediatricians online, and they're asking about the post-COVID condition, the Kawasaki-like condition. Is there anything about genetics that we can learn about why that happens? And then there are others asking about why should it be older people who are more likely to develop severe COVID? Is there something in the genetics or the epigenome that might influence that? So if we take the youngsters first, and the post-inflammation syndrome, can genetics help us understand why that happens?
Well, very probably. I mean, I think the strongest genetic effects that we've seen are in children with severe disease across the spectrum of human disease. So that's an example of what appears to be an idiosyncratic immune reaction to the virus. And we're working obviously with the Genomics England and the Genomic Study and other studies like the Diamond study to combine whatever data we can get to try to find answers to that as quickly as possible. Whether we'll find them quickly, obviously we can't know until we've done it.
I agree, and I also think that we've the patients that we've been recruiting through Genomic, there's over 3,000 now enrolled, as I mentioned earlier. In the first slightly less than 3,800 plus were under the age of 50. And there were a number in the first decade of life. So we're hoping to be able to answer that. As you know, Melita, Kawasaki's disease is thought to have some heritability and genomic architecture. We will be able to look at that. And particularly in early life, it's our intention to partner the NIHR bioresource to try and improve our ability to get line of sight on rare variants that might be causative. And we would do that by the same mechanism you and I did in the 100,000 genomes, which is to identify the parents and enroll them too, because it dramatically improves your ability to filter through what can be quite a large number of rare variants, which it might be impossible otherwise to make sense of. So we will attempt to do that. And in terms of the older adults, it's more difficult for us to say. There is a sense that genetic susceptibility in common disease is more strongly relevant for earlier onset disease. But actually, if you look at common complex disorders, the effect size that you see, the modest effect size for common variants, is often fairly constant across a life course. But Kenny and I would have to admit there must be that it's very, very likely that the possession of comorbidities reflect the age pattern that we've seen in severe COVID. So the older you are, the more likely you are to have comorbidities, the more likely you are to have ill health, and that has got to be a factor.
And might that, sorry, Kenny, go ahead.
Just on that point, so Mark is absolutely right. The ISRIC 4C study have quantified that. So Callum Semple and Anne-Marie Docherty and Ewan Harrison have very carefully quantified the effect of comorbidity. And Mark is absolutely right. It increases markedly with age. But we've recorded comorbidity in prospective case report forms, and the effect of age underneath the comorbidity is still very striking.
Yeah.
And something that's very striking about this disease, in comparison, for example, to influenza, is the apparent sparing of young children with very severe comorbidities.
So might this be an epigenetic phenomenon?
It's conceivable. So I think the other thing, I mean, I'll let Kenny talk about ISRIC's 4C's plans to do multi-omics analysis, which is the omics that aren't the genome. But just to share with you that to test that hypothesis, we will resequence a limited number of the patients with very long read forms of sequencing produced by Oxford Nanopore. This allows you to read very long lengths of the genome. And also because of the nature of the sequence, which is generated by passing a DNA molecule through a pore, you can also simultaneously read the epigenome. And you get it, if you like, for free alongside the genome. So we're going to do some limited experimentation. I wouldn't be surprised if there are epigenetic phenomena that affect outcomes.
Okay, I think we have time for one last question. And this is another popular question on the chat. Do you think we might get ourselves into a position where genetic screening across the population helps to stratify risk?
Go ahead, Mark. We talked about that a couple of hours ago.
Well, so I have mixed views on this. What I do think is we will probably identify rare variants of intermediary effect size that will be useful to describe to people that they should shield, and perhaps those will be related to some other biological marker which we measure in everyday practice. Will we get a risk score? It's conceivable because this is an external challenge, but we have to be a bit cautious about promising that, because the field of what's so-called polygenic risk scores, where you add up the total aggregate effect of genetics across a population to cause a disease, is in its infancy. I suppose if you had to choose an area where it might make most traction, it would be in a disease where there's an outside insult or trigger that is actually triggering the unmasking of that genetic effect. So it would probably be most useful there. But that's certainly something we intend to look for. Obviously, an ambition of this would be to see, are there any markers that might stratify response? Could there be people that Kenny could identify that should shield earlier and should just not re-emerge, and certainly should not go near any pub whatsoever, because it's a high risk enclosed environment for them. So if we could find that is, we were on a call where the Chief Scientist of Scotland, David Crossman, said, if you can find that, people will be very happy with you.
You get a pub passport.
Well, not just the pub passport, but the government you wouldn't have to shield. We'd be able to open the economy with greater confidence, all sorts of things emerge. But I don't think for this audience, we should attempt to promise those things now. I think that is a research endeavor, and we just do not honestly know how that's going to turn out, do we, Kenny?
Yeah, I agree.
It's just guesswork.
Well, I think we're going to have to leave it there, unfortunately. I'm sorry to those who asked questions about vitamin D and vitamin C and receptors for those, and for those who asked the inevitable questions about gene editing. But maybe we'll be able to come back later on in the month and address some of those questions to you. So thank you very much to Mark and to Kenny. That's been an absolutely fascinating half an hour. Thank you so much for your time and sharing your knowledge. As I say goodbye to you, I'd like to tell you about two other events. One is if this has whetted your appetite, then perhaps you'd like to join us on the 27th of July with the medical genetics section, where we'll be discussing some more around the genetic determinants of COVID-19 susceptibility and severity. We've got a great lineup of speakers for that. If you've had it up to the eyeballs with genetics, also at the same time is running the COVID-19 conference, sharing the international lessons learnt around the world. So we've got some international thought leaders joining us on that occasion too. So that these are all on the website. Please have a look and sign up. Please complete your survey and tell us what you thought of the webinar. And do join the RSM. It is a wonderful institution. It's really risen to the challenge of that COVID-19 has brought to it. So we very much welcome you on the team and as part of the membership. So on that note, I'm going to say goodbye, be well and be good.